Hi, everybody. Welcome back. And we are going to move forward to the second and last presentation of today's webinar with our student Ottilia Andersson, who's working at the Department of Medicinal Chemistry at Uppsala University. And she is going to talk to us about new strategies for early drug discovery and design, and in particularly looking at the examples of antibiotics, because she's at the Uppsala Antibiotic Center. Ottilia, we are looking forward to see how your projects are going and how your molecules are doing. Thank you.
Thank you, Eva. I'm gonna share my screen. That we know works. Now, you can see my screen, right?
Yes, correct. All good. And we also see the pointer, the green
Good.
pointer. Yeah.
And I remove you. Oh, you see the pointer as the blue one. That's okay. Yeah, okay. So thank you so much for having me as presenting today. And I will move a little bit away from the biofilms, which I also think is interesting, but now I will show you another part of the antimicrobial resistance research that's going on here at Uppsala University. So as Eva said, I am doing my PhD in the group of medicinal chemistry with Anders Carlén and Oskar Verho, and focusing on early drug discovery. So first today I will talk a little bit about the antibiotic research and why we need that. And then I will move on to early drug discovery and design and where it takes place in the AMR research. And my projects is a part of early drug discovery. So I will logically take you through the path of making a new drug from design of new molecules together with method development within the hit identification. And then I will finally talk a little bit about my third project, which is in hit to lead optimization of a new drug or antimicrobial resistant drug. So I am sure that we all in this environment are aware of why we need new antibiotics, and we are, especially nowadays, we have WHO who has put some of the bacterial infections as the top high causes of death worldwide, which makes us understand that we really need efficient antibiotics to treat these infections. So it's both to treat the infections that are going on and that are actually life-threatening to patients, but also we need this to prevent these bacterial infections for example, hospitalized patients, and also for patients that are going through surgery, etc. So we are really in need of good antibiotics for our patients, or for us humans. And today we are in lack of new antibacterials or molecules in the pipeline of developing new antibiotics, which we have been for several years now. Recently it was one put on the pipeline, but it's going very slow. And this is together with that we have the problem of AMR, which is antimicrobial resistant, and means that especially gram-negative bacteria develop so fast, so they develop protection to the new antibiotics that we have on the market, which makes them not useful anymore. So we definitely need new treatments. There are different ways of making new antibiotics, and here is some kind of illustration on how to. We look first at the structure, designing a structure of a molecule, test it, and see so we have activity of a molecule. Then it's important to make a good formula of it and make an actual drug, and then to put it on the market to be useful. And how do we then find new drugs? There are different ways of doing that, and today one of the common things is to look at the antibiotics we already have on the market, which is really good, and then we can use that as inspiration, and we can make some changes to the structure and see if we can have activity, and that we also have activity to the resistant bacteria. the thing with this is that it works for a while, and then the bacteria will still develop, and then it will develop a protection. So as long as we try to improve our drugs, they also improve their protection in the bacteria themselves. But with this said, all efforts is needed. So whatever we can do, and this is why I think it's so important to have this interdisciplinary view or perspective of finding solutions towards the AMR problems. So all efforts are needed. But what I also really believe is that we really need to put an effort into the early drug discovery part. So we need to find new antibiotic or new possible molecules. And why do we need these? That's because, as I said, already existing drugs, we can make changes to that. There are different mechanisms in the bacteria, and I'm sure that there are many more in this room that knows, or Zoom room, that knows more about the mechanisms of bacteria. I try to follow a bit, but I'm definitely not a pro here. So there are different things to think about when we make new molecules. We need to find a target in the bacteria to kill it. And that could be either inside the cell, in the cell wall, or even in the pumps, which is the part where if we get a drug molecule into the bacteria can have mechanisms of sending the molecules out again, which if we have the target inside the bacteria, it will be infected if it is kicked out of the cell again. So talking a little bit about drug discovery and development then, this is a known illustration of how the process is going. And the first step, which I have made green, is the in vivo and in vitro testing. And here is where we where we focus on the early drug discovery and design part, which is divided into 3 parts. First, basic research, you get an idea of what to do. And then drug discovery and design and preclinical testing. I will spend a lot of time talking about this since this is my project. This is where my project goes into. When you have good results in your preclinical testings, it's then possible to move it on to clinical testing, where you for the first time try a drug in humans. And then it's a lot of paperwork and data reviewers with the FDA revision and later on approval. Then after you put it on the market, you also have the ongoing safety testing as long as it's on the market. So here is where I will spend the rest of the presentation talking about, and I will of course connect it to the AMR research. So if we look into the early drug discovery process, it's a clear process of where we have logical process. You need first to have target identification. You need to know what you are looking for. It could be either a treatment you want to make, or you can have a target that you wanted to target in the body, or as in my case, we want to know that we can target bacteria in some way. After that, you want to find hits. So hits in this case, I mean that we find a molecule that is interesting. In some way, it's given some activity, and we have a compound that is promising for us, as we would call it. We are far, far away from an actual drug, but we have some kind of interesting molecule that we can look further into. After this, we have the hit to lead optimization, which means that we take the molecule from the hit identification part, which we know is a bit active, but nothing. And then we want to optimize this one to actually become something good. So in this part, we can look into the how the structure looks, and then compare it to results from testing. After this, when you have optimized your hit compound to a lead compound, we move on to the lead to candidate optimization. The candidate drug is the final drug that we put the most effort into, and it's like just before it's ready to be tested in humans actually. And then preclinical testings before we test it. My projects in my PhD is focusing these two parts of the process, and I think it's really nice that it's connected to both these two, and I can see the process of how organic pharmaceutical chemistry is useful in especially AMR research. So in the hit identification part, as I mentioned a little bit, is that we want to see that we have some kind of initial activity. And the main goal is to find compounds that are active in some way. And this is in general. Hit to lead optimization is also possible then to find series of hits. So here you can find different kinds of structures. You can get different ideas of, and like legs to stand on to try further on. And it's possible to make something that we call structure-activity relationship studies that I will talk a little bit about in the end. In this part of the process, we also always think about drug-like properties. So we have, since we know that we want to make drugs, we always have that in mind that the compound should be safe for our bodies. So this is always, we want it to be active, in this case against bacteria, but we want it to be safe for our bodies. So this is very important. And then as like a side story to this hit identification part, and I mentioned this because I have a project in this field, is method development, which is super important to use organic pharmaceutical chemistry even better. We still need to investigate and develop the methods that we are using to make them optimized and useful. So for this we can use different kind of technique or tools that we can develop further within the screening methods or when making compound libraries. So as I said, show this because this is how I explain my PhD times. And I have one project within this hit identification part together with my collaborator Chris. And then I have another project which is method development within that technology. And then I have a third project which is a bit different. And that is an SAR project that I will talk more about later. So digging into the hit identification part, there are many different ways of finding new hits, structures that you want to work on. And a very common one that I guess many of you have heard about is high throughput screening, or HTS, which is very useful, and it's used a lot in industry. It's automated, and the scalability it has is pretty good and big. It's usually a bit in academia, but not as much because it's due to costs and that you don't have the same capability as you have in the industry. Also still in antimicrobial resistant research, it's very common to focus on natural product screening, which means that we can extract compounds or interesting compounds from natural sources in plants, etc. And then we have a third one, DNA encoded libraries, which is more and more used, and we can have really, really large libraries where we can screen towards many different targets, for example, then bacterial targets. And we are then focusing on the DNA encoded library technology. So that's what I will talk a little bit more about. So we can make, or we are also making, a big library in just a small Eppendorf tube. And you know how small an Eppendorf tube is, but in that Eppendorf tube, in the end, we have the final library. So it's built up on that we have small molecules. So what I call as a small molecule, you will see later. But we have that attached to a DNA tag. And the DNA tag is then used both at a synthetic scaffold, which means that we are making the reactions on the DNA scaffold. But then in the end, when we have the final library, the DNA also works as an encoding system. So in the end, we can screen the library towards different targets, and we can look back into and encode it thanks to that we have the DNA tags on it, and then identify by what the small molecule is attached to the DNA. The small molecules that we react on the DNA, it's built up by different kind of building blocks. So in the rest of the presentation, I have only written BBs, so you don't get confused what it means, but that's building blocks. So the workflow, and this slide is busy, but I think it's when I go through it, it will make more sense. So we have the final library in a single Eppendorf tube from the beginning, or sorry, we make a split and pool synthesis. I come into that on the next slide talking about it. But we have the DNA tags, and we combine it with the building blocks, which you can see here. We have a DNA tag sequence, unique DNA tags, parts together with different kinds of small molecules on the other end. And in this Eppendorf tube. And when we screen it together with the targets, and we can use affinity selection, where we get information of what part of them, or which of the molecules have bound to the target binding pocket of the target. The non-binders will be washed away, and then we will do an elution process of we don't do that. We work only to make in the library, but in the end, when you have screened it, you can get information of the binders, and then you need to sequence it and identify which one or which of the compounds that have bind to the targets. So as I said, we are only focusing on making the library, which is basically this part. So the process of making this library is that we first want to have an insight of what to do, or we want to have an idea of what we want to do. And from that, we can then design, and we can investigate the chemical space. So we wanted to, design and look into the chemical space. After that, you can select the building blocks that you have of interest. So depending on what kind of library you want to make, you select the building blocks to make it as diverse as you can or as you want, but usually you don't want it to make too diverse. So it binds to whatever. You want to have some kind of idea. So you need to think a little bit about what kind of building blocks you are selecting for the process. And for that, we also have today very good help from data chemists that can investigate the chemical space in the computers for us. After that, a very important part in this process is to optimize the reactions. This is where we spend a lot of time in the lab, just optimizing and trying to get a good method for making this work and to optimize it. And this is especially important with the DNA, because we want to make sure that the DNA is intact and not affected by the chemistry conditions that we are using for the reactions. Then before you do the actual library, you move on to make a test library. And this is because it's a lot of costs of making. It's a lot of cost, and it's a lot of effort and time taking to make the library. So you want to try it on a smaller scale first. Then when you have qualify that all your steps works, we are ready to move on to the actual library. Along this time, we always need to monitor the reactions and the steps, which we do with DNA or with LCMS for oligonucleotides, and we also monitor it with agarose electrophoresis. So I mentioned this split and pool synthesis, and this means that we are working in cycles. So we call it first cycle 1, which is logical, and then cycle 2, also logical. First, we have a primer, which is the same for all the DNAs, for all the reactions, together with a DNA headpiece. So these are attached together first, same for all reactions, or for all, reactions. We do that together with a ligation of the DNA, first DNA tag. The DNA tag in this first step are 3 different tags, which gives us, when we put this in an Eppendorf tube, we have 3 different products after this step. And these are illustrated by different colors to make it as simple as possible. So this is similar to how we made a test library for our project. One single step. So when we have these different kinds of DNA tags attached to the primer and the DNA headpiece, we can then do the first reaction, which means that we are attaching one building block to the DNA headpiece, which in the other end, already here you can see, in the other end of the DNA, we have atoms. In this case, it's an amino modified version, but there are different kinds of, depending on what kind of library you want. We want to have it amino modified to make amides coupling. You will see that later. So we can make a reaction in between these ones with a new building block. We do this in 3 different Eppendorf tubes, and then after this step, we combine them into one. When we have done that, we can split them into 3 new different Eppendorf tubes, which means that in all these 3, we have the same setup of molecules. So that was the first step. So then in ligation for the next step, we always do the ligation first, which means that we attach the DNA first, and then we do the reactions. So we attach a new DNA tag to this one, and we do that with 3 different tags. Oh, this has moved a bit. And we do that with different tags in all the 3 different Eppendorf tubes, which means that we will have 3 new ones in the next step. After this step of the ligation, we can add another building block to the building blocks that are already there. So in our case, as I mentioned, we do it with amide couplings. When you have these 3 in different Eppendorf tubes, we can combine these 3 into an Eppendorf tube, a new Eppendorf tube, and then we can add another cycle of adding a new DNA tag and one more reaction. So this is basically our first project, which I have together with Chris, also a UAC student, PhD student. And our focus is to make a peptide-like DNA code library. So we have until now worked on the library design. This is why we want to have, since we want to have a peptide-like molecule, we started with a DNA or with a amino modified, as I mentioned, so we can make amide couplings in between amino acids. So we have this, this is something we buy from the beginning and it's done. And then we can add an amino acid. We can add a second amino acid, or we have added a second amino acid. And then we finally in the end add carboxylic acid, which we also can attach. to the amine of the amino acid. So after this, we moved on into validate the building blocks, which took a lot of time to know what would work. So we tried these without DNA first. And then it's important to try it on DNA, as I said, to make sure that the DNA is intact and has not get affected by the conditions we use for the reactions. Just to make sure before we started, we wanted to together with our collaborators, investigate the chemical space to see if there were more amino acids that we should include in the library. And our aim in this project is to find an antibacterial compound. We, of course, want to test it towards different kinds of target, but our main goal is to test it against antibacterial compounds that we have available. So I talked about the chemical space. This is in collaboration with our colleagues here at ILK. We use the principal component analysis, which means that we can look into several kinds of parameters at the same time. I think this is up to 12 different parameters. And it gives us an idea of how spread out the building blocks are, and that what we will include in the library. So the orange parts here, it's a bit easier when it's on Zoom than when it's on the screen. So the orange parts are the ones that we want a bit spread out. So we want to maximize the diversity of the building blocks. Doesn't mean that we want to have infinity diversity. We still want to make it, we want to have an idea of what kind of diversity we use. And this is the same again. This is, similar again. So linear approach is what we use for the synthesis. So instead of having those balls in the end, before when I was talking, I can show you here how we actually use the amino acids. So we put together, this is just for one linear approach. So we had a DNA headpiece together with the primer and the first building block. Together with this, we added the first amino acid, which I call AA1 here. And then you can see that these are a new molecule together. We ligate the next DNA tag attached to the red one, which we call the BC2. And we can then add the amino acid 2 on the other end. The same in the next step, we add a green one, which is BC3 here. And then we can, in the end, or at the last step, add the carboxylic acid. So imagine this, that we have different kind of, or we have a lot of Eppendorf tube at the same time, putting these together, run them, and then combine them and split them. So we started this with developing the conditions without DNA. So as you can see, this is just DNA look-like conditions, pretty non-harsh reaction conditions. And then we tried different kinds of coupling reagents. The one that was very successful was the Hato-Hopo-Dipea. These were not so different, but all labs have different favorite parts, and Hato-Hopo-Dipea is something that we use a lot, so we continued with that one. Worked really well on the first attachment of the building block here, as you can see. And we saw that we had a problem when we got into the attaching or ligating the second building block on DNA. So here we started a process of investigating why and how. So we tried several ideas and different conditions on this second attaching or ligating the second building block, to the molecule. When we had tried a lot, we said that we could try something else that we actually have used in other projects before, which was a new kind of coupling gradient, DMTMM. And this was successful. So we changed our process and moved on to this one. And for that, we needed to try some different ones. And just to assure that this is a process that we can use, we tried the full cycle of first we just made a DNA tag from the beginning, or had a DNA tag, and we attached all 3 building blocks just to see, so it worked, and it worked fine. And the conversions in total for all steps was above 90%. And therefore, this is the method that we used for making the library. And of course, before we started with the full synthesis of the library, we made a test library with less material. And at the moment, my colleague is working very hard on finalizing the library. So it's approaching the end of making the whole synthesis of it. And then the plan is to get to work with our collaborators. And I hope that me and my colleague can visiting and see how it's done by SciLife Lab when we screen the library. So I have talked about several times about the DNA compatible chemistry, that we know that DNA it's not super sensitive, but we need to think about what we do because it has its fragility of the molecule itself. And since we are using it as an encoding system, we really want to make sure that it's still intact. So when we started to work on these projects, we thought it was interesting to have a project in the method development process of DNA encoded library technology, just because that we also want to take part in trying to expand the toolbox. And one thing that's important in chemistry, especially in drug development, is protecting groups. It's used to protect different kind of atoms or groups in a molecule that are very reactive during synthesis. Especially here in drug development, amines are common in drug molecules. They usually are effectively used as interacting with the target protein. And this is of course also in the DNA encoded library technology. So we looked into what kind of protecting groups are there out there, and what can we investigate in the DCL. And we found the phytolamide, and it's commonly used in organic chemistry and without DNA, but not so investigated in the DCL technology. So we thought that this was interesting to try. So first, again, the idea of using DNA is first we have a bit more clear view of the whole process when we do it without DNA in the lab. So first we tried to make these reactions work, or get a method without DNA. Also here, similar to the conditions we use for DNA, of course, or same conditions, but similar molecules. So yeah, sorry, here we made a starting material first, which was very straightforward. And then we start to work on the deprotection investigation of deprotecting the phytolamide. And in DNA, working with DNA, we need water to make as a solvent to make it soluble. So the important thing here was to use water and buffer in our case. We moved on to DNA, which worked straightforward to make the starting material here. And I mean, until now, we didn't know if it would work on DNA at all. It was successful to synthesize the starting material, which is the phthalamide attached here. And then we took the starting material and tried to deprotect this one. And it seemed to work. We are still investigating a little bit here what the best method and how many equivalents of the deprotecting reagents we need. But it definitely seems to work, and we just need a little bit more time to investigate what is the best conditions for it. And this is where we are now. And after that, we will also try to deprotect it on dipeptides attached to DNA, of course. So that was my 2 projects within the hit identification field. And I also have a 3rd project within the hit to lead optimization, which is more clearly antibiotic research. Unfortunately, this is a confidential project, but I will tell you a little bit about what I am doing in the project. And I see target identification has moved. That's fine. So in this project, we got this idea from a collaborator from Brazil. He had a lot of compounds and he wanted to test. So we screened him, not me, but my collaborators at INBIM screened these compounds and found out that one had antibacterial activity. This project got into my hands and I became a part of it. And we started to look at this structure as a core structure. And we started to plan how to make a structure activity relationship study on this one. So the parts I can show is here. So we have like a core structure here underneath the blobs. And we are doing different kind of analogs, which makes that we are changing these parts and also this part to see how the activity changes. So in close collaboration with the microbiologist at INBIM or in Dermid's group, they test it and we make new molecules and we get results and we can see how the molecule and the activity is correlated and then we can test new things. So until now, we have made about 60 analogs and tested. We had promising mixed data, or we have that, and also promising or good cytotoxicity data that makes this project look good. We do have antibacterial activity both against gram-positive and gram-negative. We were pretty sure what the target was from the beginning, or we had an idea. Some of us was sure, and then we have at least had an idea. And now we are also investigating what is actually the target. So I think this is super interesting, and here we have a lot more to do, and it's very interesting. So our goal here and what we are working on at the moment is to try to improve the antibacterial activity even more. We are investigating what the target is, and also together with this, we investigate the cytotoxicity all the time to see so the drug-like properties are on track. So yeah, and of course I also have people to thank for this and to taking care of me and my projects, but especially thanks to my main supervisor, Oscar. And then I have my co-supervisors, also my collaborators, and yeah, Linus and Eva, of course. So yeah, thank you.
Thank you so much, Ottilia. Again, some applauses. So much. Oh, Grigory Grigory, did you put your hands up? Maybe he wanted to
No, I
applaud.
want to upload.
Yes, no problem. Um, yeah, so nice. So maybe if anybody has a question, again, you can write it in the chat or put your hand up. Um, Meanwhile, there was something that uh, kind of came to my mind now. I think for first time I've seen you present this work sometimes, but very early on your split and pool, like when you're making this kind of synthesis, right? That you put your DNA parts and then they ligate and then you do a reaction. So your building blocks also kind of ligate and you're doing this in steps. So first you have three different ones, then you put it all together, mix it. Hopefully you get equal amounts of all those three in these new three ependers that you use. How efficient is these are these ligation steps and these reaction steps? So you every time you put a new building block or a new part of DNA, does all the molecules that are there get this new attachment on, or how does that work?
Yeah, that's the thing. We can't know. We don't know. We have, that's why the work before is so important. So we actually try um, all the different new building blocks we do try before. So that's the only validation we can have.
So you, yeah,
Uh,
so you cannot really know if you have, I don't know, 100 of each, if they all get this new little
no,
part.
no, we only know that from how to look how the molecular mass is changing thanks to the
Mhm.
LCMS. But yeah,
And with that
that's a bad
LCMS,
sign.
how much like efficient? Because I always hear this thing in chemistry when you do reactions, you look at efficiency and you're trying to get like on the range of like 90%, right?
Mm,
Because you want everything to be converted to this new thing that you're making.
mm,
Do you guys use similar processes in this building of the library?
yes, yes, we do.
And how much is that? Does it actually work very well?
I mean, it is. This is definitely a cons with the technology. So we can only compare it to the actual like conversion and efficiency. We can only see from before.
Hmm.
Uh, so yeah, but that's yeah. I don't know. Did
Yeah,
I
yeah, yeah.
answer?
I think I understand. Yeah,
Mm,
what you're going after. And then I had another question. Or
mm,
maybe I missed this, or I don't know if it was very clear
maybe I
to
didn't
me.
say.
When you are making your libraries, you were putting a lot of emphasis. You know that it's important all the thought process before you start making it, so you decide what building blocks to use. Um, do those building blocks are chosen depending on what kind of target you are going to use after.
Yes.
So for these ones that you're making with Chris, the target is already chosen that you are going to prove them on.
Yeah, I think so. It depends on who you ask. Like for us, it was very clear. But
And
yeah,
is
you
it
know how
possibility that
people
you would
talk.
also try those libraries in completely different targets?
It might be, yeah, depending on how successful the screening is, or if we. So our goal, and yeah, it depends on the collaborators as well. But we hope that we will try it for. Now I haven't said this in so many weeks, but there are like the most common bacterial targets that LepB, LPXH, and then
Mm-hmm.
there are two more that I can't come up with now that SciLife Lab has available.
Mm-hmm.
So we hope that they can screen it towards this.
All right, I see. Okay,
But
perfect.
yeah,
Yeah.
the communication.
And those things are thought of obviously would be more successful if you actually think already from the very beginning what kind of
Yes.
targets you want to use to build this. Yeah.
Yeah. So since we know that we really would like to find a new possible antibiotic, we have looked in, that's why we want the peptide like structure.
Mhm. But that's also why it's so important to have this research that tells you what things are so essential and are so important for bacterial living, right? And then
Mm,
those can be used by the medicinal chemists to make these
mm.
kind of libraries.
Yeah,
Really
that's what
cool.
I think it's so nice with this interdisciplinary.
Yeah,
I
right.
mean, I listen to all the bacteria
You
things,
need
but
to
I've
talk
never.
to all the biologists that are studying, what are the essential things for
Yeah.
biochemistry, but biochemists as well, not only organic chemists or like medicinal chemists, but
Mm,
biochemists that understand these like essential processes.
mm,
I was just very recently learning about redox biology, how important redox systems are,
mm.
and things like that. Um, so if nobody has any more questions, some of you are still here. Just a reminder that after this webinar, we have in 2 weeks' time on Monday 19th, a very cool event that we have not arranged before at UAC, which is a UAC Career Day. So if you have not signed up for that, please go to our website and go to the link and sign up to come. We are going to have alumni from UAC coming and telling us what they're doing after graduating their PhD, and we are going to explore what are the possibilities for jobs in reality after having a PhD in such a multidisciplinary topic like AMR. So with that, I thank you all. I thank you, Ottilia, for bringing us your most updated results on your project. All of you for listening, and hope to see you at many other UAC events soon. Thank you,
Yes,
everybody. Have a great weekend.
you too.